morphology. Right? I told you to look them up. Need to know the cellular morphology. That's the shape and arrangements of the cells for Staphylococcus epidermidis, Micrococcus luteus, Bacillus subtilis, and Escherichia coli, and Serratia marcescens, because those are the 5 we've looked at in lab so far. A lot of times you'll get a question about what's the cellular morphology, what's the colonial morphology. The answer is usually easier than you think. You're thinking, oh crap, I didn't memorize the cellular and colonial morphology of all 5 of those bacteria. I'm never going to remember. The answer is in the name. If it's coccus, it's round-shaped. If it's staphylococcus, they're round-shaped in clusters. Right? If it's luteus, it's yellow. Remember, Serratia was supposed to be red pigmented? So on the Petri plate, it would have red colonies. Yours just started to produce red. It wasn't completely red. So a lot of times, the answer is already there. It's not too hard to figure out. Remember, Serratia are the red bacteria. Micrococcus luteus are yellow. Staphylococcus aureus, aureus means gold. And you saw some of them on your Petri plate. Okay, did that one. Well, I think I have the same question in there twice. With different options for the answer. Huh. Okay. Remember, we talked about fermentation. And when Dr. Pasteur was looking at wine, if the grape juice turned to wine, that was alcohol fermentation. If the wine turned to vinegar, that was acetic acid fermentation. In gangrene, the gas gangrene gives off butyric acid. That's butyric acid fermentation. When we looked at yogurt, the milk, lactose, turns to yogurt, which is tangy. That's lactic acid fermentation. You also have lactic acid fermentation in your sore muscles, right? And, okay, remember which stains are basic and which are acidic? Purple, blue, red, and green. Crystal violet, methylene blue, safranin is red, brilliant green. Those are basic dyes. Negative stains, nigrosin, which is India ink, black, Congo red, and eosin. Those are your negative stains with the acidic charge. Okay, you got to read your documents that are in Canvas, because sometimes there's questions on them that I'm going to ask on the quiz. Do you know what prebiotics are? Do you know what probiotics are?
Yeah, I'm not going to use
Probiotic is healthy bacteria. You want healthy bacteria on your body, in your body, in particular in digestive tract. So foods like yogurt that are naturally made by fermentation or even manufactured, they have healthy probiotic bacteria in them that contribute to a good, healthy flora in your body. So they actually, to some foods, add extra probiotic bacteria. But these probiotic bacteria, they need to hang out. They need fibers to hang out on. Remember the beer chips I showed you? They throw those strips of beechwood in the beer containers. Why? So the probiotic yeast, the yeast that ferments the beer, has something to hang out on, something to cling to. Same thing with prebiotics. They're fibers. You need to have fibers in your digestive tract for your probiotics to hang out on. So some foods add prebiotics also. You especially see it in dog food for some reason. They add probiotics and prebiotics to dog food. They're giving the dogs fiber and healthy bacteria. So the healthy bacteria have something to live on in the digestive tract. That's the prebiotic, is the fiber. Okay. You know what commensals are? That's your good guys. You live together peacefully, mutually beneficially, commensal. As opposed to your pathogenic bacteria, which are the ones that Might have an opportunistic infection, gets out of control. Your commensal turns into a pathogen, so to speak, or pathogens that come from your environment that make you sick. Okay, why do we even stain our bacteria? Make it easier to see them, right? We can see the shape and arrangement of the cells because we need contrast, so we stain to see contrast. Makes it easier to see the cells in our microscope if they have better contrast. That's one of the things that improves resolution. If we can see the shape and arrangement of the cells in our microscope, it helps us identify the bacteria, right? Helps us see the cellular morphology better. Okay. Oh yeah. Oh, you know what? I might not have mentioned this yet. Let's do this. Penicillin targets what?
Cell wall.
The cell wall. What specific component of the cell wall? The peptidoglycan. So if I'm a gram-positive cell and I have lots of peptidoglycan, or I'm a gram-negative cell and I only have little peptidoglycan, plus it's covered by that outer membrane, who do you think is going to be harmed more? What?
The positive.
The gram-positive bacteria. Penicillin harms gram-positive bacteria more than gram-negative bacteria. In fact, most gram-negative bacteria are resistant to penicillin because the little bit of peptidoglycan they have is covered by an outer membrane. But if you're a gram-positive cell and all you got is peptidoglycan and a lot of it, and it can damage a lot of your peptidoglycan, you're really vulnerable to penicillin. So penicillin gets gram-positive cells much, much better. It's meant for gram-positive bacteria, not gram-negative. And remember who's gram-positive. All your cocci and both of your endospore formers. Bacillus and Clostridium are your endospore formers, they're gram-positive. And all cocci are also gram-positive, so they're going to be susceptible to penicillin because of that thick wall of peptidoglycan. Oh boy. Okay, got to know your shapes and arrangements of the species and a little bit about cellular morphology. You need to know the two genera that are endospore formers. Where do endospore forming bacteria come from? The dirt, right, they're in the soil. Can you name a spirochete? What? Syphilis, good. What's the scientific name for syphilis? Remember that one. Treponema pallidum. There's another spirochete causes Lyme disease. What's the name of that? Borrelia burgdorferi. Good. Okay. Sometimes you don't know the answer to the question, but you do know what it's not. So read the question. Look at A. Could it possibly be A, or there's no way it could be A? Cross it out. Look at B. Does B answer the question, or is there no way? No way. Cross it out. So sometimes you don't know the answer, but you know which ones are not the answer. So figure it out by process of elimination. Let's say you eliminate 3 of the choices and you're down to 2. Well, that's better odds than you had before. Guess one of the 2. You see what I mean? Okay. Do you want me to write the species of the bacteria that you need to know on the board? Yes. Ooh. I'll put them here. Here's my punishment, the biohazard box. That'd be good for a hazing. Stick somebody's head in there. Well, that was cruel. I'd probably be arrested for that. All right. Okay. Escherichia coli. What shape are those cells? The bacillus, little tiny single bacilli. Bacillus subtilis. What shape are those? Bacillus, right, and they can be in chains. So it might be a streptobacillus, and they're larger than E. coli. And these could have endospores, and those wouldn't, right? And who else did we have? We had Serratia, Marcescens. Okay, does anybody remember when you did your direct stain? Serratia marcescens were tiny coccobacilli. They almost look like E. coli, didn't they?
Yeah.
These two look very similar. And then you had Micrococcus luteus. What's its other name? Luteus means yellow. A micrococcus is a staphylococcus. Sometimes it'll occur in tetrads. And you had Staphylococcus epidermidis, which lives all over your skin. I could culture it off your skin right now. Okay. E. coli on the Petri plate, translucent white. Bacillus subtilis on the Petri plate, looks like clouds. Irregular edge, grows really well, it's white. Serratia marcescens on the Petri plate. It looks white, but eventually it'll turn red. It usually has a red pigment on the Petri plate. If you take cells off the Petri plate and stain them purple, they're going to be purple. The stain overrides the natural color of the cells. Micrococcus luteus, yellow. Staph epidermidis, white, opaque white like your lab coat. White, smooth, round colonies. You can Google all of them. They have pictures of all of them on the internet. If I describe small, single, gram-negative bacilli, am I describing cellular morphology or colonial morphology? Cellular, right. If I say smooth edge with a raised center, yellow in color, that's a colonial morphology, right. Round with a smooth edge, that would be colonial. Or irregular shape, edge. So if I describe the edge, maybe the elevation, maybe the color, that's probably colonial morphology. Let me get you some props before I forget. We got 2 minutes left. Rush, rush, rush. I know. It's been a long day. I've been here for 4 hours longer than you have. Okay. Poison. Skull and crossbones. Right? These are my representatives of bacteria. When you have tiny bacilli, like the red jelly bellies, that's like E. coli and the other Enterobacteriaceae, they're tiny gram-negative bacilli. Whereas cocci are purple. The round ones are purple. Teddygrams, get it? Teddygrams? These are my Teddy Graham bears. So he's gram-positive, he's purple. Gram-positive. Negative. Red is negative. Now, it's not going to be bright red like this in your scope. It's going to be pale red. Because the cells have such a thin wall, they can't hold a lot of red. So they're going to be pale red. But I didn't know how to make them pale red. What else they got in here? Oh, here's my peptidoglycan. I'm going to need that tomorrow. This is supposed to be Helicobacter pylori. It doesn't look helical to me. It looks more like a vibrio, a curve. So if you were to ask me with a tuft of flagella at the end, to me this looks like vibrio, vulnificus or vibrio cholerae. But they say it's Helicobacter pylori. I don't know. And then somebody gave me this guy. This one is supposed to be cholera, but he's only got one